We must believe in ourselves, or no one else will believe in us. We must match our aspiration with competence, courage, and determination to succeed. Every woman's success should be an inspiration to another. We are strongest when we cheer each other on. Good morning, everyone. We are gathered here to celebrate International Women's Day, which is observed on March 8th and is preceded by week long celebrations, which are collectively known as Women's Week. A woman is so much more than just a human being. She has the power to create a life, to endure such suffering, and to consistently emerge as the strongest person in the room. My heartful greetings to all such women out there. I, Dr. Sakshi Bhartaj, on behalf of Elton Technologies, welcome you all. I am glad that I am glad to be given this opportunity. Actually, Women's Day is a day where we are recognizing, or we are giving the recognition and appreciation for all their hard work, dedication, and achievements in their own specific fields, basically. Today, I feel like wow. You know what wow means? Wow is wonder of woman. Being a woman is itself a very special thing. Women are the epitome of courage, hope, and life. I mean, it's an irony, right? To have a day exclusively for women and remind us of a good virtue of ourselves that a woman brings along. I always believe that every day is a woman's day, actually. That's the spirit we should, or we must all start articulating and impiping to our next generation as well. Let's begin with a celebration by recognizing and appreciating the female researchers through our webinar series, actually, that have achieved success in their respective areas. Indeed, to show our respect for all those women who contributed greatly to the welfare of the society and to the country consequently. However, this is the day all about valuing ourselves and realizing that one or having that full potential to do something. Altum Technologies salute all the women out there who are doing wonders all around the globe and making their nation proud. Today, actually, we have such special guest speaker with us, Dr. K. Sangeetha, presently working as Assistant Professor, Department of Biotechnologies, Savitha School of Engineering, Chennai. She had 11 years of working experience. She has done her PhD in applied microbiology from the University of Madras. She has owned more than 20 index publications, 3 Indian patents, and many book chapters to her credit. She has been invited for various talks and involved in many development programs too. She has contributed to the development of various databases. Nevertheless, she has been involved in many organizations as a committee member for the various activities. Today, Dr. Sangeetha will be going to talk about different therapeutic measures to solve the emerging and emerging viral infections such as dengue, Zika, and chikungunya with the identification and the development of their potential inhibitors. I request to all of my viewers to stay connected with us throughout the session and feel free to drop their basic queries into the chat box because we will be taking the questions and answer at the end of the session. And now, without any further ado, let's start the session. It's up to you, ma'am.
Thank you, Sakshi. I'm audible. Just want to check. I'm audible, Sakshi.
Yes, ma'am. Audible.
First of all, thank you, Sakshi, for your nice introduction towards me. Good morning to all gathered here for this webinar series. First of all, I would like to thank the organizers of Altum Technologies for inviting me to share the scientific talk on behalf of Women's Day celebrations and also specifically on the Women's Day special webinar series. Other co-women speakers are also uh, going to share ahead their views on the developments in the science. In specific, women researchers, especially the topic computational biology and chemistry, they have invited me to deliver the lecture. It's really a welcoming one. And as women, we are designed by evolution itself, evolution, and also through that is by evolution itself to take up the responsibility, additional responsibility. So evolution itself has given a chance to take up the additional responsibility. So as we are also playing the role, same thing in the organizations and also in the home, by taking the additional responsibilities. So really, it is a very proud moment for all the women for playing this, taking up this additional task in the science and also in the other avenues and other areas. So I salute all the women and also thank this Altem Technology on inviting me for this talk. Now I'll just enter into my topic. So it is actually a journey of my 10 years, 10 years work. Basically, I started my research in applied microbiology. I'm basically a microbiologist. And then when I was working associated with the Bioinformatics Infrastructure Facility Center for the first 5 years at the start of my research career, so I extended my knowledge even in the I extended my knowledge in bioinformatics, so which helped me to design a study and also to forward my research, to take my research further even in further in the bioinformatics field. Next slide please. So why we need to study about the viruses, or why we can you please go to the previous slide, Sakshi? So why we need to focus on the viruses when bacterial diseases are there, or many diseases are outnumbered in compared to the viral diseases. Viral diseases may cause acute illnesses or chronic diseases. So in a population, if you take people will be there with the acute diseases without any symptoms also, but all of a sudden an epidemic may arise to spread the infections in that particular area. So you can have the viruses with no obvious symptoms to acute infections, and also from the acute stage to debilitating infections, and also chronic infections cases, chronic infections carriers like in HCV, HIV cases, and HBV cases. So what are the symptoms? General symptoms of a viral infections are mostly diverse and in specific for the particular diseases like fever, diarrhea, encephalitis, human immunodeficiency, tissue damage, and organ failure. So coming to the lethal infections are caused by Ebola viruses, Marburg. Next slide. Can you please go to the next slide? So viral infections or even originated from, that is organ related, like eye infections, herpes simplex viruses, adenovirus, cytomegalovirus mostly causes the eye infections. Mumps virus causes paratitis, and myelitis are caused by polioviruses, HTLV, and Coxsackie Bay viruses causes pancreatitis, and common cold is caused by rhinoviruses, parainfluenza viruses, respiratory syncytial viruses, and encephalitis or meningitis by this JC virus, measles, adenovirus, and rabies. In addition, virus papilloma virus, Merkel virus, and other viruses are also associated with different types of cancers. So cancers, that is viral infections, are also one of the agents to cause the cancer in the human beings. Coming to the other significant viruses which causes the human health problems are human immunodeficiency virus, hepatitis B virus, hepatitis C virus, which causes the liver damage and acts as a chronic carriers in the patients. Next slide, please. Why cataloging the human viruses is challenging if you take, because symptomatic also, and it is completely cleared from the patients from within 5 days of infection, and in the other viruses it causes chronic infections. So we have to first categorize which type of viruses, and it is also fully based on the patients. It may cause acute infections and also chronic infections. So in few cases what happens is it may remain dormant for a longer period of time and may show reactivation only after 10 years or even 15 years after it spread in the human body. Next slide. Next slide, please. So still a major gap is existing in determining which viruses are contributing to this chronic illness and also to the chronic diseases. Sorry, please, previous slide, please. Sakshi, previous slide, please. So because all the, and it is also involved in the host responses. So bacterial infections, it is not the case in the bacterial infections where the bacterial, where the host mechanism or the host role is very limited. But in the case of these viral infections, the host role plays a very important factor because all the viruses are intracellular pathogens, or otherwise they require the host machinery for its utilization and also for its progeny production. So what happens during the system, during the course of the diseases, it stimulates the immune system and acts as the drivers of systemic inflammation, and also influences the immune homeostasis. So it disturbs the overall immune homeostasis, or the general homeostasis of the human beings. And also the specific sequences of the viral infections has an impact on the human function and overall health. And selected viruses or a few viruses causes greater morbidity and also susceptibility in the human beings. Next slide, please. Next slide, please. So we have totally about 200 human viruses so far, but only nine diseases are treated with the licensed antivirals. So what are the viruses like HBV, hepatitis B viruses and hepatitis C viruses are treated with this. So coming to this emerging and re-emerging viral diseases in India, we although we have these 200 human viruses, in India we are having only a few viruses that are persistently causing infection and mortality and morbidity in India. Coming to the viral hepatitis, we have these cases like A, B, C, four types is causing, and it is circulated in the Indian population. 4% of people is affected by this disease. And coming to this enteric fever, rotavirus, noroviruses, which causes 20%, and especially the children are affected with this disease. Dengue is causing about 10% of the patients or of the population is infected with this dengue hemorrhagic fever and dengue shock syndrome. Chikungunya 4%, and another arthropod borne diseases like Japanese encephalitis and West Nile virus is 2%. Measles and rubella again it is 16%. Chickenpox 14%, and the Crimean Congo hemorrhagic fever and Casno forest disease and Zika virus as 1%. And we have the other minor non-viral diseases, which accounts to about 29% of the pop, disease in the population. Next slide, please. Next slide. Sakshi, next slide, please. So these are the uptrends in the viral diseases. What are the uptrends in the viral diseases is first earlier in this, that is before freedom and before freedom in 1947, or also even before 1980s, we had the increased amount of measles and poliomyelitis in India. But now that trend has gone down. We have limited amount of when compared to this chikungunya, dengue, and Japanese encephalitis cases. Now these cases are reported more when compared to measles and poliomyelitis. Next slide. Next slide, please. So among that, only 15% the vaccines. Other for the other things, we need to make. Even though overall efforts has been put, we need many. We need to develop therapeutic avenues have not been stemmed for these things, and increasing trends of the global disease burden and geographical expansion emphasizes us to or to work towards the therapeutic avenue areas. So coming to that estimation of these diseases, about 70% of these emerging infections of humans are zoonotic in origin, and two-thirds has been originated in wildlife. In coming to this Indian population, we have this chikungunya again, dengue, Zika virus, Japanese encephalitis virus, and chiasnul forest as diseases as the main diseases. They all belong to the family of Flaviviridae, Alphaviridae, and Nairoviridae family. Next slide, please. So arboviral infections such as chikungunya and Zika and also dengue, they are the three circulating viruses which co-circulate together in the mosquitoes and causing the menace or causing the morbidity and mortality in India. So once this arboviral infection, why it is called as the re-emerging viral infections is chikungunya once it was reported in 1963 and 1973. After a gap of 40 to 50 years, it reappeared in the year 2006 with the virulent strains circulating in the population and affecting a large scale population in a major epidemic cases. So even Zika, we are witnessing the re-emergence of Zika in India, and a few states it is reported like Madhya Pradesh, Gujarat, everything. The earlier outbreaks was with the Asian strain of chikungunya virus. Now, after 2006, the Southeast Asian African strains are circulating India. Next slide, please. Next slide, please. So these are the two major vectors, which is aegypti, Aedes albovectis. These are the major vectors in Indian Ocean region, and in the African countries, the other vectors are also involved, like Aedes falciparum, Aedes tyleri, Aedes luteocephalus, Aedes africanus. And why this? What caused this mutation, or what caused the virulence in the strain of Southeast Asian Africa or Southeast African strain when it re-emerged in 2006 was the mutation that was noted in the 226 amino acid position in the E protein of chikungunya virus. So there was a mutation. Instead of alanine, valine was replaced in the 226 position, which caused the viruses to be cholesterol independent and made the viruses to propagate very easily and spread and also the virulence capacity of the viruses increased during the re-emerging condition. Next slide, please. So these are the geographical distribution. We have basically basically three clades are based on the genetic differences in the E protein, envelope protein. We have three things like West African, East Central, South African, and Asian clades. And these are the cases that has been reported from India in the last 5 years. Still now we have the cases of about 1,93,000 cases. We are still reported in 2022. So still we have the chikungunya cases. It is being circulated and it is present in the Indian population. Next slide, please. Next slide, please. So, although it doesn't cause the morbidity, why we have to concern or why we have to worry about these diseases? So, we all know that chikungunya does not cause any morbidity or the significant morbidity. No, sorry, mortality, not mortality. Then why we have to worry about these diseases? Because this diseases causes the permanent inability in the patients, that is inability to move easily, and arthralgia is the major concern of this disease. So during that 2006 epidemic, about 25,588 disability adjusted life years were calculated due to these diseases, and also the productivity loss of 39.1 crore were estimated due to these diseases. Next slide. Next slide, please. Next slide, please. So immunopathogenesis, it is not only the viral factors that is involved in the disability or involved in the pathogenesis, also the immunological components that of the host also plays a major role in and the decrease in the rantis level in the patients. These are also the factors that causes the immunopathogenesis in the due to in the chikungunya patients. Next one, next slide, please. So treatment and management, if it, for both the chikungunya, they are treated symptomatically with the antipyretics like acetaminophen, aspirin, and non-steroid anti-inflammatory drugs. And also this chronic arthritis now it is treated with chloroquine phosphate, corticosteroids, and other disease-modifying anti-rheumatic drugs, and even tumor necrosis factor blockers. And anterior uveitis in case if eye is infected. Uh, Topical steroids and cycloplegic agents are given. And the ocular hypertensions are also treated with these hypertension blockers only. So, what is the current status of development of vaccines for chickpea? In case of chickpea, all these things are under different phases of clinical trials. So, various vaccine candidates are under trials or under trials like live attenuated vaccines and formalin inactivated vaccines, subunit vaccines are available, or there under the production, and also under different stages of clinical trials like preclinical and clinical phase 1, and nothing has been so far licensed for the or approved for the treatment to the patients. So, what is the common thing between these diseases or the differences between these three diseases is that these three diseases co-circulate in a single patient. So, at a time, in case a patient can be infected both by chikungunya virus and dengue virus also, or by dengue virus or Zika virus also. So, all these three viruses are prone to cause the co-infections in the patients, and they are circulating. They co-circulate even in the mosquitoes or co-circulating in mosquitoes also. So dengue and Zika belongs to the Flaviviridae family. We have in the dengue 5 serotypes. The fifth serotype now they have found out based on the envelope protein differences. And on Zika virus and dengue viruses mostly share their homology. Homology in the sequence structure, in the sequence amino acid structures, they are sharing the homology to 90%. And incubation period of dengue virus is about that 3 to 14 days and days. So next slide, please. So we have the fevers for the chikungunya as fever, headache, pruritus, pain, myalgia, arthralgia, maculopapular exanthema, conjunctivitis, retroorbital pain, hepatomegaly, lymphadenopathy. Even for this Zika virus, we have this microcephaly and Guillain-Barré syndrome. Dengue is categorized mostly as dengue shock syndrome and dengue hemorrhagic fever. So normally, dengue will be causing a mild fever in the asymptomatic patients. It may progress in the severe conditions to dengue hemorrhagic fever and dengue shock syndrome. So, what are the vaccine developmental attempts to dengue? Whether we do have vaccines for dengue? So attempts to develop the vaccine against dengue had failed many a times. Ota and Kimura in 1944 they formulated this formalin inactivated vaccine, but the attempts failed. It was unable to neutralize or induce a larger amount of antibodies in the patients, in the treated, in the subjected or the treated patients. Similarly, but Sanofi Pasteur came up with the vaccine dengue vaccine in 2015, but these has been licensed only to individuals to the age of 9 to 45, but these are also unable to neutralize the heterotypic type of infections. Only to the homotypic infections it is able to neutralize. When it comes to the secondary heterotypic infection, it is only providing, or it is only showing a transient protection level to those type of patients. Next slide, please. So, what is the necessary for us to develop these type of necessity for the development of novel drugs to these arthropod borne diseases or viruses? Is first, we don't have any licensed drugs so far. We don't have any licensed drug or the licensed drug for the licensed drug for the treatment of this dengue, chikungunya, and Zika. And secondly, is that we don't have licensed vaccine even for vaccines for chikungunya and Zika virus. And we have the burden of immunosuppressed patients of 3.25 renal transplantations is done per million in India. So we have to take care of these patients not by getting infected with any other viral diseases. And also the sanitation and quarantine measures to control dengue has not been achieved so far with desired objectives. We are unable to eradicate this mosquito population. So, and also malnutrition children are increased, and also due to the increased proportion of people with immunocompromised due to the poverty levels. So we need to design the strategies to develop drug. So this drug development approaches is important. The drug development approaches, novel drug development approaches is required, like direct acting antivirals and host directed antivirals, because direct acting antivirals may sometimes result in the higher toxicity, and also the clinical effectiveness is not achieved, or then the treatment levels it is not achieved to the 100% level. So we need both the host directed designing of host directed antivirus and also the direct acting antivirals, which could reduce, the combination of these could reduce the risk of viral resistance and also increases the chance of pan protective effect, which could be able to protect from all the serotypes and from the all the clades of the viruses. So we can design the virus inhibitors at many levels from the fusion. From the fusion level, we can take as a fusion we can take design the entry inhibitors, and we can go for the fusion inhibitors. At the translational level, we can go for the translational inhibitors. And in the host, we can also target the host components like glucosidase inhibitors, which is required for the maturation and the folding of the proteins in the viruses. So when we target these inhibitors of the host, it would result, or it would suppress the production of the matured virions. So we can target this host inhibitors and autophagy inhibitors of the host, and also the other nuclear transport inhibitors. So we can design the drugs either targeting the host or either targeting both, targeting the viral proteins. So we have the druggable targets like druggable targets. There are many druggable targets that is available for these three viruses. What are those? Are capsid proteins. We have envelope, which comes under the category of structural proteins. And in the non-structural proteins level, we have non-structural protein 1, non-structural protein 2, non-structural 3, 4, and 5. We have different 5 types in the dengue and Zika, and we have these 4 types in the chikungunya virus. So in our journey of this for 10 years of research, we have screened the libraries belonging to the natural phytochemicals, synthetic chemicals, repurposed drugs, and peptides. Now we have entered into the peptide research. So why these targets, and among these targets which we can take or which we can give an higher priority, is that based on the genetic variability and the knowledge gained from the other viruses and also the viruses, we have the targets which could be given the higher priority, like NS3 protease, NS5 polymerase, and E glycoproteins. And also nextly to this other capsid proteins, NS4B, NS3 helicase, and NS5 methyltransferase enzymes. So there is a high degree of conservation of the amino acid residues with the structural functions in specific to the glycine, proline, and cysteine among the Flaviviridae family. So when we target these type of conserved proteins, when we target these type of conserved proteins that is present in the envelope protein of this Flaviviridae family, that could be used as a broad spectrum antiviral agent for the treatment of the diseases. So and also the, can you please go to the previous slide? Sakshi, can you please go to the previous slide, please? So we also have these catalytic residues that is present in the methyl transferase genes, and also these catalytic motifs in the NS3 proteins, protease, and ATP and RNA binding regions of NS3 helicase. These are found to be very, very highly conserved. So once if I am designing a drug for the dengue virus, that could be sure it may 99% or 95% possibly it would work out for the Zika virus also. So these are the genomic or the structural organization of the dengue virus. We have this capsid, membrane, envelope, NS1, NS2A, B, NS2B, NS3, NS4A, NS5B, NS5. These are the structural and non-structural organization of dengue virus. And coming to this chikungunya virus, we have this into 4 categories like NSP1, NSP2, NSP3, NSP4 for the non-structural. Structural again is divided into C, E3, E2, 6K, E1. So NSP1 is involved in the negative strand RNA synthesis, NSP2 helicase protease, NSP3 RNA synthesis, NSP4 RNA dependent RNA polymerase. In the case of dengue, it is NS5 which functions for this, which codes for this function RNA dependent RNA polymerase function. So this ED is the envelope dengue protein is here, dimer, and it is located at the interface between ED2 from one chain, and it forms an ohm. Next slide, please. So Zika virus shares the about 90% homology with the envelope protein of dengue virus. Similarly, here the genomic arrangement is very similar with that of the dengue virus. Like C capsid protein and pre-M, it is having envelope protein NS1, 2, again A, B, NS3, NS4, NS5. So these could be taken as a druggable targets, which we can work on to produce or to find out the drug molecules. Next slide. So envelope proteins of dengue, in which E2 envelope domain of 2 is very, very important. Envelope 2 is involved in the fusion of the viruses with the host, where it falls between the region of E2 52 to 132 region and 193 to 280 amino acid region of the envelope protein, which this yellow part depicts the fusion involved in the fusion of the dengue viruses. So this dengue virus forms a beta barrel domains. Envelope protein of dengue virus forms a beta barrel domain, which is especially this envelope dengue 2 region is highly conserved. It is found to be highly conserved, and 3 is involved in this cellular adhesion and viral uptake. Envelope protein the domain 3 is involved in the cellular adhesion and the viral uptake. These, as a whole, it forms the homodimers, and they join together to form the rafts that spans the capsid of the viruses. Next slide, please. So we have the non-structural proteins. Non-structural proteins are equally functioning well, equally functioning, which is very important. So targeting any one of these sites also will be helpful in developing the drugs. NS1, if you take, it is involved in immune evasion, pathogenesis, and viral replication. NSP2A is involved in the viral RNA replication complex and also in the virion assembly, and these also are involved in the host immune responses, and NS2B is involved in membrane destabilizing activity and forms the viroporins. NS3 is a protease enzyme, NS3 serine protease, which is involved in the autocleavage and cleaves the polyprotein at dibasic sites. So similarly, 4 and B is very important in the NS3 because it is involved in the phosphorylation process. Next slide, please. So these also we have the ample of targets for the viruses like structural and non-structural proteins. Altogether, we can have at least 9 druggable targets for that. Although we prioritize 3 or 4 of them. Coming to the host targets, equally we have the knowledge that has been used to study the viruses and the interactomics with the host has made us to gain knowledge and to identify the host factors that is contributing in increasing the viral progeny production and also the pathogenesis of the viruses in the host. So we can also target the host factors or the host receptors or host things as the inhibitors. So main thing is this alpha glucosidase inhibitors. Alpha glucosidase inhibitors. In that alpha glucosidase inhibitors, like immunosugars like castornospermine and 1-deoxynitromycin has already been worked against these dengue viruses. They have already made a wide research how this able to inhibit this glucosidase, which is involved in the viral folding and maturation. So this is found to be working on this, and it is under clinical trial for this castornospermine. And coming to the other nucleoside biosynthesis inhibitors like IMPDH inhibitors, this one is example is a ribavirin. Ribavirin now particularly treated for the HCV viruses as a standard drug, and other cyclophilin inhibitors, and nuclear transport inhibitors, peptidyl prolyl isomerases, and cyclosporin is an example for that, which is now under trial or which is used for the treatment. Next other host inhibitors, or we can target HSP70 heat shock protein and DDX3 and polyamine synthesis inhibitors. Also other inhibitors like this lipid related processes in the inhibition of acetyl-CoA carboxylate and fatty acid synthase. So these are a few host factors. They are also contributing in the increased pathogenesis of the host and also in the production of the viral progenies. So we can target these host factors also. Next slide, please. So even though we are screening through the bioinformatics software against these targets, at the last stage we need to study these viruses, whatever the compounds that we have screened, whether it is working in a live against these live viruses through in vitro studies. Then we can go for the clinical studies. So in vitro studies are assays that is available for the flaviviral compounds, or many assays are available, like several cell-based phenotypic assays and the target assays are available. Cell-based assays, if you take these live viruses, like we can use directly the live viruses in assessing our compounds based on the CPE and the plog assays. And or as otherwise we can go for the direct use of these VRPs, subgenomic viral replicons, or also the virus-like particles for conducting these in vitro studies. But the first one, if you take this live viruses, either the CPE or it is a time-consuming, laborious process, and we need the biosafety cabinets also. But in the second level, we don't need the biosafety cabinets. But the thing is that there is an restriction level of entry in these type of second cases. Or directly we can go for the target assays using the enzymes and also these fluorescent linked proteins. Next, next slide, please. So far it was just an overview of how the viruses, what is the general or the structure of the viruses, and what are the druggable targets that is available for that, and also the assays that is present to understand the efficacy or the workability of that compounds in the lab level. So now I'll be shifting on to my my research. Or so far, how many libraries or how many compounds that we have found out in my journey of 10 years. So against this dengue virus, we worked about 5,000 phytochemicals, and for against this NSB, NS2B, NS3, NS5, 7,000 compounds we worked on. We screened about 5,000 for the envelope and 7,000 for this NS2B with the natural phytochemicals, and synthetic chemicals more than 6,000 and more than 6,000 for both the envelope and NS2B and NS3B and 5. And coming to this chikungunya virus, we screened of about 6,050 compounds for against this envelope protein of chikungunya virus, and same 6,050 against the NSP1 of chikungunya virus, and 8 plants and 24 plant extracts. Zika virus, NSP1, we screened about 5,550 compounds, and NSP3, 5,550. Chemical category, we screened about overall to this dengue and Zika virus 6,000. And repurposed drug 10, we screened for this dengue and chikungunya. Peptides 2,000, and now we are screening for this peptide, only specifically for the envelope. We have started up this journey to the peptides. Now, next slide, please. So screening of phytochemicals. So we categorized for our study or our research into 4 screening of phytochemicals and the synthetic chemicals, and next is that repurposed drugs, and next last is the peptides. So coming to this phytochemicals, why we need the phytochemicals when the synthetic chemicals are available is, it would when we discover this phytochemicals, or if a phytochemical is working out, means it would give an it would give an expanded and a full stretch result. That is, it would give a benefit. It would be highly beneficial because it will give a one-touch solution. Okay, it would give a one-touch solution to the problem when we are screening the phytochemicals, and if that phytochemical is working on that, which could be massively or extensively used in the population for their treatment. But in the case of the synthetic chemicals, it involves that we need to again analyze it and we have to synthesize. But it is not the case or with the phytochemical compounds or the plant extracts directly if you are doing it. So first, this is our one of the study, and which got published by after doing that is spectral characterization, antiviral activities, in silico admet and molecular docking of the compounds, isolated from the Technona grandis to Chikungunya virus. Next slide, please. Next slide, please. So among that, we isolated 5 compounds, and which was compared with ribavirin, which was was compared with ribavirin actually. So it showed an high selectivity indices. So compound 2 and 3 showed an increased selectivity indices from 136 to 156. And compared to the ribavirin also showed 529, but it doesn't work for the African isolate actually. It doesn't work for the African isolate. So, but our compound, when it was subjected, it worked for the African isolate and produced about 14 to 11 in selectivity index. In comparison with that compound 4 also produced an high selectivity index of more than 4. Coming to this mutagenicity and carcinogenicity analysis that was done through this pre-admit analysis, carcinogenicity it was negative. That is compound 3 and 4 was found to be protective in nature, and it doesn't exhibited any mutagenic properties and all. So also the distribution, it had a very good plasma protein distribution and absorption level also for the compound 3. Next slide, please. Next slide, please. So next is that we took the two plants and their compounds and docked against these structural and the non-structural proteins of Zika viruses. And when it was so these two medicinal plants, and also the quantum chemical parameters and density function theory and ADMET was also assessed for this. So as a end, at the end, we were able to found out that a few compounds they exhibited a very good interactions, and also exhibited a very prominent interactions like 10 compounds out of the 32 compounds we subjected. 10 compounds showed promising interactions. It actually showed promising interactions like 1, 2, 3, 4, 6 penta orthogalloyl BD glucose and bacopside 3, 4, 5, geranine, gualic acid, all those things interacted with the Zika target very well. And the quantum chemical parameters of these compounds were also estimated based on the three components, and that the polar molecules were able to dissociate better than the non-polar molecules, and that was represented by the dipole moment. The dipole moment is also an index of lipophilicity and the ability of the drug molecule to cross the various biological membranes. And the third, the smaller energy gap was observed. The smaller energy gap also observed for the few compounds. And also that they exhibited a very good interactions and expressed the satisfying admit properties. And acid and bacopside 3, bacopside A found as an potential inhibitors of the Zika virus protein targets in our study. Next slide, please. Next slide, please. Can you please go to the next slide, please? Next slide, next. You can skip these slides. This has been explained. So these are the hydrogen bond interaction profile of the compounds against the Zika virus protein targets. Envelope protein domain 3, NS1, NS3, and these are the varying interactions to this active site of the active site. Next slide, please. Next slide. Next slide, quantum. Okay. So these are other work was that molecular docking and antiviral activities of the plant derived compounds against Zika virus. In this, about we screened 5,550 phytochemicals. 5,550 phytochemicals we screened, and through collaboration with the Spain, with the other collaborators, we tested this compound also. We tested the compound also individually for the antiviral activity also, antiviral activity. So in that compounds for screening through this, we did this through flexible docking analysis in Discovery Studio. We screened overall 39 compounds against this NS3 protein, and against the envelope protein, we came with the conclusion of 22 as the it compounds of the lead. Next, with the final compounds we screened were about it came out around 10 with the higher binding energy at the active site. So what are those is muricatetrosins and neoandrographolites, eleutheroside B, bicalin, and all ellagic catechin and panoxinins, and 5 compounds against these envelope proteins: epigenin, glycopyranoside, bicalin, and isonimolinisaccharide and madicassic acid. And those were again subjected to ADMET. Among that ADMET protein and envelope 4, we came out with the first again subjected for the in vitro significant activity to the Zika virus. Next slide, please. Next slide, please. So in this, we use this NS3 protein and envelope protein as the main two targets to study these viruses. NS3 protein now as both has got a NS3 protein has protease function, and also envelope has the function in the entry of the virus. So we targeted that. In NS3, we targeted mostly the helicase. Prominent sites in the NS3 which we targeted was helicase between the domain 1 and 2, between the domain 1 and 2, and in the junction domain 1 and 3 for the envelope protein were targeted. And also the sites that were close to the extra pyrophosphate density region, and also in the RNA binding groove, where the polar and hydrophobic characteristics were highly present, those regions we targeted. We targeted against this NS3 and the envelope protein of these viruses. In this, eleutheroside B interacted with the ATP binding site, and can you please go to the next slide, please? So in this eleutheroside B interacted with the ATP binding site and in the near proximity with the pyrophosphate, spanning the amino acid residues of arginine 462, 309, 201, glutamine 286, all those things. And next, also muricatetrosins, neoandrographolide, and sangeetha A extended hydrophobic interactions at the ATP binding sites. And the other two compounds, catechin and epigallocatechin, are formed specifically hydrogen bond interactions with the amino acids near the pyrophosphate group. Epigallocatechin, muricatetrosins, they all formed this interacted with the amino acid residues present in the P loop. Whereas this bicalin, canthrine 6, and ellagic did not form any interactions in the P loop, which is found to be highly conserved and an active site in the envelope protein of Zika viruses. And also this homo-lumo results were found to be promising for these compounds, and also that they also exhibited desirable pharmacokinetic properties by ADMET. So when it was taken to this antiviral activities, except this madicassic acid, all the other 3 compounds exhibited a very promising antiviral activity. Next slide, please. Next slide, please. Next slide. Next. Next. Next slide. So these are the electrodensity clouds around the molecules that has been tested here. So the high density or influenced to high densities of the homo region are influenced to electrophilic attack, and lumo region are prone to this nucleophilic attack. Next slide, please. So these are the antiviral activity results and also the cytotoxicity results of the compounds that has been tested in collaboration with the other laboratories. In Spain, we collaborated and they reached out to help us with these compounds and activity for assessing the activities against the Zika virus. So coming to this cell viability and that is on cell viability titers, and these compounds eleutheroside B and epigenin and neoandrographolide grapholide were found to be non-toxic even at 1000 microgram when it is extended, but they were non-toxic at 1000 or 100 micromolar concentrations. But madic acid, it was toxic at 10 micromolar itself. So even in the antiviral activity assessment, they exhibited the antiviral inhibition of the antiviral growth at 100 micromolar by eleutheroside, epigenin, and neographylide compounds. Next slide, please. So next is the again antiviral activity of the other compounds that was for Zika virus. So for whatever, firstly I discovered that was discussed about the Chikungunya virus progress, how much we reached, and the Zika virus, how many compounds were screened for the Zika virus. Next was is it is against the dengue virus. We screened for the dengue virus again using compounds 7,000 phytochemicals using astrogalacide, that is among that we found out the activity from astrogalacide 2, 3 and 4. Next slide, please. Next slide. So we screened 7,000 phytocompounds. Among that, 10 phytocompounds were found to be highly effective. And in that, astrogalacide 2, 3, and 4 were found to be with a good activity exhibition in compared to the other standard compounds like, next slide, please, ribavirin and SAM and also the BOG pockets, BOG compounds. So BOG compounds and SAH standards ribavirin were compared for these assessments. Next slide. Next slide. So they also exhibited higher interaction profiles. Next slide. Next slide. Next slide. Next, you can skip. Next is this exploring the potential anti-chikungunya virus activity of the phytocompounds. Here also we screened specifically for the chikungunya virus 6,050 compounds. This was again taken up to the further study, antiviral studies. Next slide. In vitro, again in this astrogalacide 2, 3, 4 were found to be effective and played a potential role in the inhibition of the viruses, chikungunya virus, wherein 3-TRK was used as the protein targets. Next slide. Next slide. So coming to this assessment of this anti-chikungunya activity, it was tested for both the African clade and the Asian strain, both the strains. Previous slide. This both the Asian strain and African strain, it was tested. It was found to be effective for both the Asian and the African strain of chikungunya virus. So coming these are those, so these are the data, these are the things that has been screened for from the phytochemicals so far from our research. Coming to this synthetic chemicals and small molecules, we went for the screening libraries like PubChem and Zinc database. Next slide, please. INO's library of compounds. So among these, we found we have scrutinized about 10 to 13 compounds that is found to be effective from a library of about 6,000 compounds. That we are that's still under the subjection and subjected out to in vitro studies. and we came with the fruitful results for the synthetic chemicals also, a few synthetic chemicals also. Coming to this repurposed drugs, among the repurposed drugs we tested, we got the results, good results for tetracycline, doxycycline, and suramine. They interacted with the envelope protein and also with the NS3 5 of dengue and chikungunya virus combinations. Tetracycline and doxycycline individually, they exhibited a very good result at the in vitro studies level for the Asian and chikungunya ECAS strain of chikungunya virus, and also for the serotypes, different serotypes of dengue viruses. And in the combinations also, they exhibited a very good promising results. Even at the level of 15.62 microgram per ml, doxycycline and ribavirin and tetracycline and ribavirin exhibited the results. Next slide, please. Next slide, please. So these are the results that we got in the laboratory. Next. So as a result, we been granted 3 Indian patents for the novel pharmaceutical combination of doxycycline and ribavirin. Patent we got 3. Next slide, please. So now we have entered into the screening of peptides. So far, the compounds that has been tested for dengue, balapiravir is the first nucleoside analog, which is a prodrug of 4-azidocytidine. But so far, although it had exhibited good efficacy, it has failed and not reached the market due to the toxicity and also the decreased potency in treating the patients with human peripheral blood mononuclear cells. So dengue, as well in specific, if we consider dengue is infecting the cells, but over in specific the human peripheral blood mononuclear cells. So only when it is infecting this human peripheral mononuclear cells, our drug, whatever that we want to target, that also should be able to interact with the human peripheral blood mononuclear cells and could be able to eliminate the virus from that part also. So that's why that balapiravir, even though it has been put under the clinical trial, it was unable to go to the market. It was able to reach the market and it failed. The drug failed. Similarly, the other drugs like chloroquine, still now it is used as an urgent treatment for this chikungunya virus and dengue viruses. And prednisolone is used, but it is not effective in the dengue hemorrhagic fever and dengue shock syndromes. And the other drugs like these are few drugs like lovastatin and selgovicin, or these are a few drugs that are under clinical trials, but they have failed due to the inability to reach or to interact with the human peripheral blood mononuclear cells, especially with dengue. So there are many challenges and opportunities that are available in this field, like although these compounds, the screened compounds that work at the in vitro level in a particular cell lines, when it comes to work in the, that is when it is to interact with the important components of cells like human peripheral blood mononuclear cells, there is a failure in these type of results. So we need this. So whatever the compound that we are identifying as the lead compounds, that need to be tested in a different panel of human cell lines. And next thing is that many assays like to assess the liver efficiencies, three-dimensional organoid development models. So these are challenges that are open to the newcomers where they can extend their knowledges, and cellular thermal shift assays to identify the changes in the drug development, and development of more animal models is required besides AG219, and non-invasive assays to measure the inflammation levels of the diseases like F-fluoro, deoxyglucose and PET, and also that stabilizing the polar surface area and to restrict the compounds even in the screening level itself. So these are many of the challenges and opportunities that are available in the drug discovery processes for chikungunya, dengue, and Zika viruses. And the upcomers can take up these as a challenges and also can extend their knowledge in that. So I hope. Thank you. Next slide, please. Thank you, Sakshi.
Thank you so much, ma'am. It was very informative session. So these we all know about these common disease condition, but the permanent inability these disease condition are going to build for a human being. It's lifelong. So still there is a gap in between for the development of new drug-like candidates, which could be the hit molecule for these kind of diseases. As ma'am very well explained that how we can utilize the homology modeling techniques to develop new inhibitors, novel kind of drug molecules, while utilizing these in silico prediction or the GAD models. As ma'am very well explained, how she has explored the flexible docking of the Discovery BioVia's Discovery Studio, how we can utilize these like homology modeling techniques, then optimization of a ligand molecule, how we can use these pocket active sites of the protein, like ma'am has utilized this viral envelope proteins for the development of inhibitors. So yeah, yes, there is a gap, but still the challenges are there, but we can cope up all these challenges with the with the saving our times and the money by using these softwares like in silico predictions and the softwares like discovery BioWay Discovery Studio. Yes, we have some questions, ma'am, actually. So okay, it's coming on the screen. How do we use the natural products or study the inhibition of viral drug targets?
So natural products, we should know the structure of the chemical compounds first. We have to isolate, and we have to characterize through X-ray diffraction or the NMR studies. We have to first characterize the compounds that is present in the plants or whatever the product that you are using first, and you have to elucidate the structure. With that structure, you can go for the targeting. So you can use the different targets against these viruses.
Okay.
And if it is found to be of promising, if it doesn't have any what if it is having good admit properties, means then we can go for the in vitro studies further in the lab using different cell lines like viro cell lines or HUH.
The next question is, on what basis screening of candidates were done mentioned in the slide screened libraries?
See, we didn't go very specific with that. We chose four categories. As I told you before itself, we chose four categories: phytochemicals, one category, then already available databases, other category. Third category is that repurposed drugs we went, and fourth one is peptide. Now we are searching for that. Now we are working on the peptides from the different databases.
Okay. So the next question is
Yes, sure. You
also,
can find the interaction between. Yes, sure. You can see the interaction between the drug target and the compounds. First you should identify the active site, and what is the site that is where you want. Where is the conserved site, active site, where you want to work upon. First, you have to identify. Then you can make it to interact at that site, and you can explore where which amino acid it is interacting, whether it is interacting at the ATP binding site or the helicase binding site. You can go for that. Yes, sure, you can see the interactions.
adding to the ma'am, actually, we in Discovery Studio, if we are doing the interaction in between the ligand and the protein target molecule, so we are getting the interaction scores, basically the binding scores in the minus C Docker
Yes, yes.
energies and C Docker interaction energy. So yes, we can definitely check for the interactions in between the ligand and the protein molecule. Okay, so the next question is, what is flexible docking? How will it be helpful? Okay, it's a nice question.
See, we have that flexible docking is that wherein you can use the different types of sites, okay, different types of target sites to where the compounds can work upon that. It is not like rigid, you are not fixing at a particular place to work on that. So where you can work on the different types of active sites, based on that you can we can categorize.
Yes, basically the flexible docking, if adding to the ma'am, so it is like a flexible. Usually we are rigid the protein molecule and we make the ligand molecule as a flexible. But in the flexible docking, we making both the component as the flexible, so that we can get the more interaction by exposing more binding sites of a protein and different configuration of your ligand molecule as well. So next question. Okay. Okay, so I think we have done with our question and answer session. So thank you once again, ma'am, for answering the questions. It was very informative session, and it was pleasure to have you with us. So thank you all. Thank you. It was a grateful that. You could join us today, and as a part of our week long celebration, we hope to see you all there in our forthcoming sessions. Of course, we will be having a quiz round during our sessions, and you can get a chance to win the excited gift vouchers. So stay tuned with us. Let's do it together. Thank you. Thank you, everyone. Thank you so much.
Thank you.